Good morning to all. Welcome to the Diamond Group of Institutions, Neet and K-Cet online classes. As today's class, sexual reproduction in flowering plants is a continuation of yesterday's. So in the previous question, some important one. First one is point of the odd one. Point out the odd one. Here, they given as a four options. In the four out of the four options, which one is option is odd one? We have to identify. So they given as nucellus, embryostat, micropyle, and the pollen grain. See, nucellus it's a tissue. It is helpful for storage of food materials. It's helpful for storage of the food materials, and it belongs into the female reproductive part or female. And the embryostat is involved into the fertilization, double fertilization. So embryostat also the female reproductive part. And micropyle. So when we see the structure of medasporangium or ovule, a small opening, their integuments covering or integuments are absent. That is we call as a micropyle. So this is the micropyle. So this is also belongs into the female reproductive structure only. And coming to the pollen grain only male. It's a male reproductive part. Pollen grain is a male gametophyte. It's involved in a fertilization. So out of the given four options, A, B, C belongs into the female reproductive structures, and pollen grain one only belongs into the male reproductive structure. So answer is D. Pollen grain odd one is a D. And next question: formation of gametophyte directly from sporophyte without meiosis. Formation of gametophyte directly from the sporophyte without meiosis. It's called as options are given as apospory, apogamy, and parthenogenesis and amphimixis. Amphimixis means through the sexual reproduction, and parthenogenesis is a type of asexual reproduction. New individual it was developed. So here question asking gametophyte from the sporophyte. Sporophyte is a diploid structure. Gametophyte is a haploid structure. So formation of gametophyte directly from the sporophyte without meiosis means at the time the gametophyte also will be don't present in the form of a diploid only. So that condition is called as a apospory. And apogamy, what's the meaning of apogamy? Formation of sporophyte from the gametophyte. Formation of sporophyte from the gametophyte means in a reverse. Here gametophyte is directly from the sporophyte. So sporophyte to gametophyte, it's called as a apospory. And here gametophyte to sporophyte, it's called as a apogamy. So question is asking as a sporophyte, formation of the gametophyte from the sporophyte, it's called as a apospory. From the sporophyte in the gametophyte is developed, it's called as a apospory. So answer is a apospory. Apogamy means formation of the gametophyte from the formation of the sporophyte from the gametophyte body, it's called as apogamy. In the next question, which ones produces androgenic haploids in anther culture? So which one they can produce the androgenic haploids in anther culture? Anther culture, it is application of tissue culture. It is used into the production of haploid plants. So out of the given options, which of the following options are they are used into the production of anther culture? Option 1, option A, anther wall. Anther, it's a diploid, so anther wall also diploid. Tapetal layer of anther wall. Anther walls are they contain as a epidermis, endothelium, middle layer, and tapetum. These are the four layers are diploid only. So tapetal it is a diploid. Otherwise, we are also called as polyploid condition. And connective tissue. So in the center of the anther, connection between the anther and the filament, that connecting tissue will be not present. That is also the diploid. and end pollen grains. Pollen grains are developed by the process of meiotic divisions. Now the pollen grains are haploid. So which ones produce androgenic haploids? So haploid culture in a tissue culture will be going to used to the pollen grains. Pollen grains are we are going to use to the production of haploid plants. So answer is D. The next question, generative cell was destroyed by laser, but normal pollen tube was still formed because. So you can see structure of the pollen grain. It's having two layers, outer exine and inner intine. So here these are the pollen grain contain as two cells, generative cell and the vegetative cell. So this is the vegetative cell and this is the vegetative nucleus. This is the generative cell and this is the generative nucleus. So question it is, generative cell was destroyed by laser. So now these are the generative cell, it will be done destroyed. And pollen tube was still formed because here functions are the uses of the generative cell and vegetative cell. So the generative cell or generative nucleus again undergoing as mitotic division and form into two male gametes, or we are also called as sperms. And vegetative cells are, it's a large in size, is helpful for storage of the food materials, and these vegetative cell is involved in the formation of pollen tube. We are destroying the generative cell. Here we are not giving any harms into the vegetative cell. So that's why vegetative cell it was present in live condition, and these vegetative cells are due rise into the formation of pollen tube. But generative cells are there involved into the fertilization, that is syngamy and the double fertilization. We are also called as triple fission. Syngamy and triple fission is involved as a double fertilization because of the involvement of two male gametes. Here we are destroy the generative cell means these are the male gametes are not formed, means it's not involved into the fertilization, but pollen tube is developed from the vegetative cells because vegetative cell is involved into the formation of pollen tube. So vegetative cell is not damaged. Option B, content of tilled generative cell is stimulate the pollen tube pollen growth. Laser beam stimulates growth of pollen tube, and the region of emergence of the pollen tube is harmed. If the region of the emergence of the pollen tube is harmed, means it's not helping to the formation of pollen tube. And here we are destroy the generative cell, then also pollen tube is formed because. So here we are destroy the generative cell at the region of emergence of the pollen tube is harmed, means automatically the pollen tube will not formed. And here laser beam stimulates the growth of pollen tube. Laser beam does not stimulate the growth of pollen tube. Boric acid and sugar solution is used into the stimulation of the pollen tube growth. And content of tilled generative cell stimulate pollen growth. Content of the tilled generative cell, here the water structures are the tilled, they are not stimulate the pollen tube growth, and remaining as a option A, vegetative cell is not damaged. Pollen tube is formed from the vegetative cell only. We are not give any harm into the vegetative cell, so automatically pollen tube is formed. So answer is A. Yes. Next question. There are 10 flowers in one individual plant of Frutillaria. In each microsporangium of every stamen of all flowers, there are 30 microspore mother cells. So how many pollen grains are formed from the plant? So here they given as one of the plant name it is Frutillaria. So hint for of this question, the Frutillaria plant it belongs into Fabaceae family. So characters of the Fabaceae family. Every flower it contain, each flower contain 10 stamens in the form of 9+1 arrangement, diadelphous stamens. So you know there are 10 flowers. Number of the flowers are 10, and each microsporangium of every stamen of all the flowers there are 30 microspore mother cells. And how many pollen grains? So here 10 flowers are there present. 10 flowers, each flower is contained as a 10 stamens. So number of the stamens are 100 stamens. So and other one of the hint it is, and these are the 100 stamens. When we see the structure of the stamens, so stamens are having filament and anther. So now these are the anther. So the anther will be going to contain as a 4 theta. So 100 stamens are there present. These are the 100 stamens. All are belongs into the dithecous anther. So these dithecous anthers are 100 stamens that contain as a dithecous anthers, and each one of the theta they contain as a 2 pollen sacs. So each one of the anther they contain as a 4 pollen sacs. So 100 stamens into 4. Total number of the pollen sacs are 400. Total number of the pollen sacs are 400, means 100 stamens are there present. These are the 100 stamens are equals into 400 pollen sacs. So now each one of the pollen sac is contained as a 30 microspore mother cells. So now the 400 pollen sacs are there. So that each one of the pollen sac is contained as a 30 microspore mother cells. 400 into 30 will be that as a 12,000 pollen mother cells. So 12,000 microspore mother cells will be donated. And now how many pollen grains are formed? So here 12,000 there belongs into pollen mother cells, means each one of the pollen mother cell can give rise to 4 pollen grains. So now 12,000 into 4 will be that as a 48,000 of pollen grains. So now this plant it belongs into the Fabaceae family. It's contained as a 10 flowers. The 10 flowers are equals into 100 stamens. That 100 stamens are they contain as a that 100 stamens are all belongs into the dithecous tetrasporangiate anthers. So that's why number of the sporangia of these 100 stamens are 100, and each one of the microsporangia means pollen sac is contained as a 30 microspore mother cells. So that's why we'll be getting to the 12,000 pollen mother cells. So now these 12,000 pollen mother cells are undergoing as a meiotic divisions and producing to the 48,000 of the pollen grains. So number of the pollen grains are produced from the Crotalaria gentiana. It's a dicotyledonous plant and Fabaceae family. 9 plus 1 stamens are there present. So total number of the pollen grains from this plant it will be there as a 48,000. Hint it is Crotalaria gentiana belongs into the Fabaceae family. It's contained as a 9 plus 1 stamens. Total 10 stamens are present in each flower. So that's the concept. And the next question, in angiosperms, all 4 microspores are tetrad. Microspores of tetrads are covered by a layer which is formed by. So now you see at the time of similarly single pollen mother cell can undergoing as this is the pollen mother cell can undergoing as a meiosis and can form into pollen tetrad. This is the pollen tetrad. And these are the pollen tetrads are covered by a presence of a layer. That layer it is formed by options are cellulose, cellulose, sporopollenin, and petocellulose. Petocellulose is just chemical. It was seen in a pollen trimer, and sporopollenin is present in outer layer of pollen grain, and cellulose is a cell wall of plant cell, and callose. So here the 4 pollen grains are pollen tetrads are they covered by a presence of a layer. That layer we call as a callose layer. So answer is a callose layer. This callose layer it will be going to degenerated by the presence of callase enzyme. It will be going to disintegrated by the callase enzyme. It was developed from the tapetum. Tapetum can produce the callase enzyme. Function of this callase enzyme can degrade the callose layer. Later, these pollen tetrads are they can separate individually. They can form into individual pollen grains. And the next question: which one of the most common embryo sat in flowering plant? Which one is the most common embryo sat in flowering plant? Embryo sats are we know that is a female gametophyte. Most common type of the embryo sat in a flowering plants are. So they given as a monosporic 8 nucleated 7 cell structures, bisporic and bisporid they're given. So you want to know the difference between the monosporid and bisporid type of the female gametophyte development. So here the monosporid means one of the nucellus tissue can act as a mesospore mother cell. That nucellus tissue of the mesospore mother cell can undergoing as a reductional divisions and form into mesospore tetrad. So out of these four mesospore tetrads, we call as four haploid mesospore tetrads. So three are degenerate, one only functional. That is we call as a FMC cell, functional mesospore cell. It's a haploid. Now you see development of the embryo sat. It was developed from the single cell. This is the FMC cell and undergoing as a 7 mitoid division and can produce into the 7-celled 8 nucleated structures. So that is we call as a female gametophyte. Otherwise, we are also called as a embryo sat. That female gametophyte or embryo sat is developed from the single functional mesospore. It's called as a monosporid type. Monosporid type. All of the angiospermic plants, most of the angiospermic plants, they can produce the embryo sat from the single mesospore. So that's why it is called as a monosporid type. That monosporid type of the embryo sat is contained as a 1, 2, 3, 4, 5, 6, 7. 7-celled 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleated structure. So monosporid type of the embryo sat it is contained as a its embryo sat is having 7-celled 8 nucleated structure. So monosporid, 8 nucleated and 7-cell structure. Answer A. Then what about the bisporid structure? So here in a similarly at the time of the development of female gametophyte in a bisporid manner, nucellus tissue can give rise to one of the nucellus, that is mesospore mother cell, and this mesospore mother cell again undergoing as a meiotic division and that can produce into the mesospore tetrad. Out of this, here 3 cells are there degenerated, but here 2 cells only degenerate. 2 cells are there functional. So now these are the 2 cell structure. They're involved into the formation of 7-celled 8 nucleated structure of the embryo sat. We call as a bisporid. So answer for the most common type of the embryo sat of the flowering plants is a monosporid, and it's contained as a 7-celled and 8 nucleated only. A is the answer. And then when ovule is curved and embryo sat becomes horseshoe shaped, ovule is called as. So here ovular body will be going to curved and the embryo sat is becoming as a horseshoe shaped structure. That is called as. They given as options. Amphitropous ovule. Amphitropous ovule means, so these are the ovular body, it will be going to curved. Ovular body, it's ovular body, it was curved, and this embryo sat, this is the embryo sat, it was becoming as a horseshoe shaped structure. So this is the horseshoe shaped structure of embryo sat. This type of the embryo sat is called as a amphitropous ovule. And what about the cisnotropous ovule? So cisnotropous ovule means in these are the cisnotropous ovule, that finicle is completely curved, the finicle is completely curved the mitropyle. So here this is the mitropyle, the entire mitropyle is covered with the finicle. It's called as cisnotropous ovule. And compylotropous ovule. Compylotropous ovule means here the ovular body, it will be done curved. It's near into the finicle, the mitropylar, and then finicle that is less distance will be done presence. So this is we call as compylotropous ovule. Orthotropous ovule. So ovular body and mitropyle and finicle both are there present in a same radius. This is called as a orthotropous ovule. So amphitropous ovule means ovular body will be going to becoming as a horseshoe shaped. In between the mitropyle and the finicle, less distance will be going to present. Those are not very close. They are present in close only, not as a very close. If that very close condition it was present means here ovular body and micropyle. Both are their presence as very close to condition. This is called as a anatropous ovule. But the question not asked from the anatropous ovule, it's from the amphitropous ovule only. So answer is A. Question. Match the following ovular structure with post-fertilization structures and select the correct alternative. They given as a options ovule, finitil, nucellus, and polar nuclei, and match with endosperm, aril, seed, and perisperm. See now, ovule is also called as a metasporangium. After the fertilization, means after fertilization, this metasporangium or ovule becoming as a seed. And option B, funitil. Funitil is a start of ovule, and these are the funitil after the fertilization can act as a aril. It's light as a third integument. Nucellus. After the fertilization, nucellus tissue is helpful for storage of the food materials. Persistent nucellus, it's called as after the fertilization, it's perisperm, and polar nuclei. So in a female gametophyte, central cell is contained as a two nuclei. Those are called as polar nuclei. After the fertilization, those it can form as a endosperm. So now A, ovule is paired with seed. B, finitil is paired with aril. A3, B2. A3 and B2, and C, nucellus, it pairs with the perisperm, C4. A3, B2, C4, answer is C. And the next question, who is author of book Introduction to the Embryology of Angiosperms? So the book Introduction to the Embryology of Angiosperms, book written by options are Maheshwari, Birbal Sahini, and T.M. Mahabale and J.S. Singh. So out of the given scientist names, Maheshwari, Panchanan Maheshwari wrote the book of Introduction of Embryology, and Panchanan Maheshwari is called as a father of embryology. Answer A. And option question number 11, embryo axis above the protolidon is called as. So at the time of seed germination, they can form as a, they contain these structures. This is the embryonal axis. So embryo axis above the protolidon. This part is called as epiprotyle, and this part is called as a hypoprotyle. Epiprotyle is formed into a shoot. Hypoprotyle is formed into a root. So question asked as embryo axis above the protolidon is called as, this is the protolidinary node above the protolidon, it's called as epiprotyle. It will be going to form into the shoot. They are given as epiprotyle, hypoprotyle, finitil, and the raphe. Finitil, you know, and the raphe represent as finitil, extension of the finitil. It was attached into the ovular body. Where the finitil is attached into the ovular body means extension of the finitil is called as a raphe. This part is called as a raphe. So this is the finitil. Junction between the finitil and that ovular body is called as a hilum. And next question. The diploid number of chromosomes of angiosperm plant 24. The number of chromosomes in the pollen grain and the endosperm and integuments will be. See now, if the plant diploid chromosome number is 24, we have to identify the chromosome number in the pollen grains. Pollen grains are their presence as a haploid condition by the process of meiotic division from the anthers. By the process of meiotic division, they can produce the haploid pollen grains. If the diploid plant body is 24, means haploid will be get as a 12. And endosperm. Endosperm is a nutritive tissue. It is a product of double fertilization, and here in that endosperm, endosperm chromosome number should be, so diploid chromosome number is 24 plus 12 haploid will be get as a 36. In a endosperm is a triploid. And again they are asking integuments. Integuments are the protective layer of the ovule. So these integuments are, and metasporangium or ovule is having diploid condition. So diploid condition is a 12, and integuments are also will be drawn to, sorry, diploid condition is a 12, integuments are also having as a 24. So 12, 36 and 24 answer D. Symphonodemy in angiosperms. Symphonodemy in angiosperms means, so here option A, tube-like male gametes. Option B, motile male gametes. Male gametes produced in a tube. Male gametes carried in a tube. So symphonodemy, it was started from the gymnosperms. So from the in the gymnosperms, pollen can produce the pollen tube. That pollen tube can carry the two male gametes. That male gametes are that can move through the pollen tube. Such type of the condition is called as symphonodemy. It was started from the gymnosperms. So male gametes are carried in a pollen tube. Answer is D. Male gametes are produced in a tube, and tube-like structure of male gametes or motile male gametes are we are not considering as a symphonodemy. And nuclear endosperm. Nuclear endosperm is nuclear endosperm has, it's not a nuclear, it should be nucella. Nuclear endosperm has, after the fertilization, in the angiospermic plants, centered endosperm, it will be done forming by the product of triple fission that can form into endosperm cell. Primary endosperm nucleus. This primary endosperm nucleus can undergo many number of cleavage divisions and form into the new, that is endosperm. So that nuclear endosperm has many number of the divisions will be done to take place. So they given as a every nuclear divisions followed by the wall formation. Here, these all of the primary endosperm nucleus can undergo the divisions and form into multinuclear conditions. Here, wall formation is absent. They given as a wall formation. And option B, initially free nuclear divisions followed by the wall formation. So they given as a initially free nuclear division. Here it is also called as a free nuclear endosperm. So these are the free nuclear endosperm. Firstly, karyokinesis takes place. Many number of nuclei is formed. Multinuclear condition it will be take place. After that, the nuclear endosperm is converted into the cellular endosperm in a coconut that can form into the wall formation. So initially free nuclear divisions followed by the wall formation. And option C, first division followed by wall formation and other free nuclear. So first division can followed into the wall formation, it's called as cellular endosperm. So out of the given three options, every nuclear division followed by the wall formation is also character of cellular endosperm, and that is the first division followed by the wall formation is also the character of cellular endosperm, and initially free nuclear divisions followed by the wall formation, that is we call as a nuclear endosperm. Answer it is B. The next question. In a monocotyledonous seed consists of one large shield-shaped cotyledon, it's called as. In a monocotyledonous seeds, a shield-shaped large cotyledon is called as. Option A, aleurone layer. On the surface of the seed or below the seed coat, a layer will be present. That a layer it is helpful for storage of the food materials in the form of a protein. That is called as a aleurone layer. It can store the food materials in the form of a protein. And scutellum is a large shield-shaped structure we call as a scutellum. Answer B. And polyptile, upper part of the in these are the monocotyledonous seed that is shoot tip, it will be covered by the presence of a cap-like structure. A cap-like structure on a tip of shoot, it's called as a polyptile. Hilum is a junction between the finicle and the ovular body. So a shield-shaped structure in a monocotyledonous plant or monocotyledonous seed, it's called as scutellum. And the last question means next one, pollination by wind is called as. Pollination by wind is called as. Options are transferring of the pollination through the wind is called as. They given as xenodemy, Anemophily, autogamy, and none of this. Geitonogamy, it's a type of pollination, means it is that is structurally self-pollination and functionally cross-pollination. And anemophily, transfer of the pollen through the wind. Here the wind or air, it can act as a pollinating agent. Autogamy is a self-pollination. Both are we are going to depend as a self-pollination only. So answer it is anemophily as a wind pollination B.